Welcome all to our best practices for topical generic product development and ANDA submission webinar. For those of you who might be rejoining us again from yesterday's webinar, welcome back. And for those of you just joining, we are very glad to have you here with us today and hope to have another great session. My name is Lieutenant Nora Lim, and I am with the Small Business and Industry Assistance, or SBIA program, which operates within the Division of Drug Information in FDA's Center for Drug Evaluation and Research's Office of Communications. It is my pleasure to once again be your SBIA host for today's webinar. Just some reminders and notes again before we begin. This session is being recorded. It'll take us a few days to process the recording, but you will soon be able to access it from SBIA's webpage. You may notice the link on the right where it says download the presentation. Note that this link here is only active during the real-time presentation. The final survey is the mechanism by which you will receive a certificate of attendance, which will support CE credits for RAPS, SOCRA, SQA, and ACRP. This certificate must be downloaded immediately after completing the survey, otherwise the opportunity is lost. Even if you do not want or need a certificate, please take a few minutes to complete the evaluation, as your feedback will help guide the design of our future events. Also, we invite you to visit our Cedar Learn webpage, where you can find links to SBIA tutorials along with other educational resources, many of which offer CEs. Please note that the evaluation and certificate are only available for 2 weeks, which means that the deadline date is August 25th. The link will be posted at the end of the event. Here, we've included our QR codes to access SBIA on our social media channels. Stay connected to learn about upcoming webinars, conferences, and regulatory information, and also to find recordings of past events. You can scan the QR codes with your phones and be taken to the appropriate platform. And here is a listing of our upcoming SBIA events. Please visit our fda.gov/CEDAR SBIA website for more information on each of these events. Now it is my pleasure to introduce our moderator for this webinar, Dr. Priyanka Ghosh. Dr. Ghosh is acting team lead in the Division of Therapeutic Performance 1 in the Office of Research Standards within the Office of Generic Drugs. And now I'll turn it over to Dr. Ghosh.
Hello. Thank you everyone for joining us, and thank you Lieutenant Lim for the kind introduction. As Lieutenant Lim shared, I'm Priyanka Ghosh. I'm an acting team lead for topical products within the Office of Research and Standards, Office of Generic Drugs. And on behalf of our entire team, it is my pleasure to welcome you to today's webinar. Now let me talk about what we hope to achieve with the webinar today. Over the last decade, within the scope of the GADUFA Regulatory Science Research Program, we have invested significantly to develop more efficient bioequivalence approaches for topical products applied to the skin. Such efficient approaches are now established for prospective generic products, reflected within product-specific guidances, and are widely used by the industry. Prospective generic applicants also have the opportunity to talk to us about such approaches within the scope of product development meetings. And in vitro studies that can often support a demonstration of bioequivalence within the scope of such approaches include comparative physicochemical and structural characterization tests, or Q3 tests, in vitro release tests, or IVRT studies, and in some instances an in vitro permeation test, or IVPT studies. We have hosted multiple workshops and presentations over the last few years to discuss these approaches with you. And today, we would like to continue to discuss these types of studies in some very short presentations. However, what we want to achieve with this webinar for the majority of the time is to focus on answering any questions you may have related to these approaches or methodologies, and how you can successfully utilize them to support a demonstration of BE for your prospective generic product. The panel sessions are scheduled after each of the 3 talks that are scheduled for today. And you can submit your questions within the Q&A, and I will be posting them to the panel. We would also like to thank all of you who took the opportunity to submit your questions during the registration process. That provided us an opportunity to triage the questions, and we will be posting them to the panel as well. With that, it is now my privilege to welcome our first speaker for today, Dr. Sam Raney. Dr. Raney is a thought leader in topical and transdermal drug products with over 30 years of experience in skin research, producing numerous research manuscripts, review articles, book chapters, and patents in pharmaceutical product development. Dr. Raney has been a researcher and adjunct professor within academia, a principal or sub-investigator on over 400 pharmaceutical product studies, has led senior management roles within the industry, and serves on multiple expert committees and panels for the United States Pharmacopoeia. He is currently the Associate Director for Science in FDA's Office of Research and Standards and serves as the Chief Scientific Advisor for topical product bioequivalence issues in FDA's Office of Generic Drugs. Dr. Raney holds a bachelor's degree in molecular biophysics and biochemistry from Yale University and a PhD in biochemistry and molecular biology from the University of British Columbia in Canada. So now I'll turn it over to Dr. Raney for his presentation.
Thank you. Thank you, Dr. Ghosh, for that very kind introduction. It's my pleasure to spend a few minutes with you today. Today's all about helping you to develop topical generic products. Toward that end, we've published numerous product-specific guidances over the years with efficient characterization-based approaches in many cases. And I'm sure those of you who are familiar with those PSGs have noticed that those that include characterization-based approaches often have very specific recommendations for which Q3 attributes to characterize. The question then becomes, for those products that don't have a published PSG, I want you to come away from today knowing which Q3 characterizations would be relevant for your prospective generic product. And the other thing I want you to come away with is an understanding of what bioequivalence approach would be appropriate for your product. So I'll offer the standard disclaimer here and begin by saying that we're going to talk about the concepts of Q1, Q2, and Q3, predominantly Q3. Q1, of course, is the components, the ingredients in a topical product or in any product. Q2 refers to the composition of that product in terms of certain percentage of ingredient X, another percentage of ingredient Y, and so forth, that quantitative formulation composition. But Q3 is what describes the arrangement of matter in a topical product or in any product. And Q3 characterization provides a profile of physical, chemical, and structural attributes that is quintessentially characteristic of that product. Now, when we talk about Q3 characterization, we're typically referring to a collection of specific tests that can be performed to describe different aspects that collectively describe the arrangement of matter in ways that are useful because they relate to how the product will perform under clinical use conditions, and also useful because these Q3 characterizations enable us to systematically compare different aspects of the arrangement of matter between a generic product and its reference product. So the first type is the characterization of appearance and texture. This is the look and feel of a product. Also of its phase states. Does it have globules? Does it have suspended particles? And the structural organization of matter. We basically want to understand the architecture and the interactivity of that molecular machinery of the system, because this is not going to be apparent from the dosage form naming, the nomenclature. For example, the structured matter may be organized as an emulsion, but the dosage form may be a topical gel, and that's fine. It may be a gel. Similarly, two creams may have a different particle size distribution, but that doesn't change the fact that they're still the same dosage form. They're still both creams, although they're not necessarily bioequivalent. Now, if there's a suspended drug, we want to characterize the molecular form of the polymorphs that are present because the rate at which the drug is released from the formulation may be different for different polymorphs, and we want to characterize this within the drug product. Also, the rheological behavior tells us a great deal about how the excipients interact within the molecular machinery and how the system responds to stress. So this typically involves using a rheometer to characterize the flow curves across a range of shear rates, although at the very least you want to characterize the apparent viscosity at different shear rates like low, medium, and high. And the water activity or drying rate is incredibly important to characterize because differences in water or solvent activity or drying rate can directly alter the formulation composition, the thermodynamics, and the bioavailability of the drug. Now, pH can also have an enormous impact on a variety of characteristics of the formulation, whether it's the viscosity of a gel or the ionization state of the drug, and pH is dependent upon how well the product is buffered. So it's important to characterize the pH of products with aqueous formulations and to characterize any buffer systems as well. Now, let's talk about oleaginous components and components in oleaginous formulation. So for products that are comprised of more than about 70% oleaginous contents, like many petrolatum-based ointments, it's typically feasible to characterize the product itself using the tests that are listed in the USP monograph for petrolatum and to record the actual quantitative test results. And from what I've seen, in addition to those quantitative tests, comparing the relative proportions of hydrocarbons in a given product and comparing that between a test and a reference standard product can be one of the most meaningful and actually useful characterizations to tell you something about the performance of the petrolatum-based ointments. And then this characterization of specific gravity, this is important to ensure there are no major differences in the amount of entrapped air that may have gotten mixed into the formulation. And finally, characterization of metamorphosis-related changes, because not only is the product undergoing evaporative metamorphosis, but it may also be subjected to shear forces that may alter the bioavailability through the microstructure. So it can be important to characterize the influence of dispensing the formulation from a tube versus a pump, for example, if the product has multiple packaging configurations. And also, metamorphosis may occur as a function of aging. So it can be important to characterize Q3 attributes at different points in time across the shelf life of the product. And when you're comparing multiple batches of test and reference products, just be mindful of the age matching as closely as possible, because those different batches are going to be different ages. Now, among this entire set, we think about these first three as being basic Q3 characterization. And we think about the entire set as representing comprehensive Q3 characterization. Now, it's useful to think about it in this way because you can describe a dosage form using basic Q3 characterization. And this is relevant to any single product, for example, the reference standard product or the prospective generic product. You can perform basic Q3 characterization on that product alone. Or basic Q3 characterizations can be used to compare both the test product and the reference standard product to demonstrate that both products are the same dosage form. Similarly, to support a demonstration of bioequivalence, comprehensive Q3 characterization can be used to compile a detailed profile of Q3 attributes that specifically describes the nature of that individual product, the prospective generic product or the reference standard product, and to describe the arrangement of matter that may modulate the systemic or local availability of the active ingredients from that product. But likewise, comprehensive Q3 characterizations can also be compared for both the test product and the reference standard and can demonstrate that there's no significant differences in Q3 attributes between the two products. Now, doing this substantially mitigates the risk of potential failure modes for bioequivalence that would otherwise arise from any differences in Q3 attributes. Okay, so by this point, you have a really good idea of how Q3 characterization can be utilized to understand the arrangement of matter in a given product or to be able to compare the arrangement of matter between products. The reason that this is such a powerful thing is because it becomes the foundation of understanding what bioequivalence approaches a particular generic product may be eligible for. And I'd like to describe this in terms of the thinking that goes behind current PSGs that are available for many products, because the same thinking would apply even in instances where a product-specific guidance doesn't exist for a product that you would like to develop. And the question that you would begin with is, for your test product, your prospective generic product, is the arrangement of matter the same as in the reference product, at least within the range characterized for the reference product? If yes, then the next question is, well, is the underlying matter the same? In other words, is there any difference in the components and composition? If there's no difference in the component and composition of the prospective generic product and the reference standard product, in other words, no difference that would significantly affect local or systemic bioavailability, then that prospective generic product would generally be eligible for the characterization-based approaches that are frequently described in current product-specific guidances. Conversely, if the arrangement of matter is different, the Q3 profiles of your prospective generic product and the reference standard product are quite different, then it would be generally eligible for traditional in vivo bioequivalence approaches that are often also described in current PSGs. What becomes, I think, a particularly interesting scenario to consider is when all the Q3 attributes, the entire profile of Q3 attributes, are well matched between your prospective generic product and the reference standard product. In other words, the arrangement of matter would be considered to be the same, but that there is some small difference that may significantly impact local or systemic bioavailability. What happens in these situations? And I think this is a really valuable place that we can talk a little bit more about in the upcoming question and answer session, about the types of approaches that would be appropriate, which would generally focus on beginning with all of the evidence utilized for a characterization-based bioequivalence approach, and then specifically looking at what those differences are and what the failure modes are for bioequivalence that arise from those differences, and what evidence would be appropriate to mitigate the risk of those specific failure modes for BE. Now, you may say, in order for the underlying matter to be the same, doesn't that mean that the components and composition have to be Q1 and Q2 the same? What's the distinction with this no significant difference criterion compared to Q1, Q2 sameness? Well, the NSD formulation assessment criteria are based upon the principles for assessing Q1 and Q2 sameness, but they also take into account certain differences that have previously been determined to be acceptable based upon evidence. So certain minor differences in components and composition may be acceptable either based upon information available to FDA or information submitted in an ANDA. Specifically, information demonstrating that there's no significant difference in the local or systemic availability of the active ingredient relative to the reference standard product. So this may be shown, for example, with an IVPT study or perhaps a plasma PK study showing that the minor differences don't affect local or systemic bioavailability. Now, in relation to that reference standard product, which the generic product is compared in a bioequivalence study, in the approaches that bioequivalence is based upon a characterization-based bioequivalence approach, then the characterization of the formulation is really the bioequivalence approach. It's part of the bioequivalence approach. And therefore, since that bioequivalence study is conducted in comparison to the reference standard product, the formulation of the prospective generic product is also compared to show that it has no significant difference compared to the reference standard product. And this is distinct from the way that Q1, Q2 assessments are always done in relation to the RLD. Because for a topical product, the reference standard, the current reference standard, may not be Q1 or Q2 the same as the RLD, and that RLD may in fact now be discontinued. Now, according to 21 CFR 31494, an ANDA, like for example, maybe the current reference standard if it's not the RLD, can include different inactive ingredients compared to the RLD as long as the applicant identifies and characterizes the differences and provides information demonstrating that the differences don't affect the safety or efficacy of the proposed drug product. And so this continues to be a key part of that bioequivalence approach. Now, another way that this no significant difference formulation assessment sometimes allows for some differences is in the ingredient grade. So for example, the reference standard may contain a specific grade of an ingredient like carbapol 934P or petrolatum USP, but the prospective generic product may be considered to have no significant difference if it contains minor differences in the ingredient grade, like using carbapol 974P instead of 934P, or white petrolatum USP instead of petrolatum USP. Likewise, the assessment of subcomponents may mean that while the reference standard may contain a proprietary ingredient that's already kind of a pre-blended mixture containing specific quantitative amounts of subcomponents. So, 30% subcomponent A, 30% subcomponent B, 40% subcomponent C. Well, the prospective generic product formulation may be considered to have no significant difference if it contains those same quantitative amounts of each subcomponent instead of using that pre-blended proprietary ingredient. And similarly, instead of this situation, if you have the RS using a proprietary ingredient that's a pre-blended mixture of variable quantitative amounts of subcomponents, in other words, 30 to 70% of subcomponent A, 30 to 70% of subcomponent B, in those situations, that prospective generic product can be considered to have no significant difference as long as it contains quantitative amounts of each subcomponent within those ranges that were found acceptable for the reference product. Now, another aspect of this is the form of the ingredient. So the reference standard may contain an ingredient like EDTA disodium that can exist in a dihydrate form or an anhydrous form, where the test product, the prospective generic product, can be considered to have no significant difference if it contains a different hydrate form of the ingredient with adjustments for the quantitative amount of that pure ingredient and the water component, so that it ultimately ends up having the same components and composition. Also, ingredient purity can come into play. So the reference standard may have an ingredient like alcohol USP, which can either be 73.5% of 95% alcohol or 72.57% of a more pure 96% alcohol. Now, similarly, that test product, that prospective generic product, can be considered to have no significant difference if it basically contains the same amount of alcohol, ultimately of pure alcohol, but using a different purity of the original alcohol with adjustments for the diluting factor of the water, for example. And then when it comes to colors or fragrances, the RS may contain a very specific color or fragrance, and the prospective generic may be considered to have no significant difference if it contains a different color or fragrance, as long as it doesn't affect the bioavailability of the active ingredient and doesn't affect the safety of the drug product. And the pH modifier. This is one that I think that most people are more familiar with, that often you have a single point nominal amount, name only, of a pH modifier in the reference standard composition table. And this may not reflect the actual quantitative ranges that were determined to be acceptable for the reference standard, or that maybe the reference standard specifies in its composition table that there's a quantity sufficient to achieve a target pH for that reference standard. So that prospective generic product can be considered to have no significant difference in pH modifier amount if it doesn't contain the same exact nominal point value amount of pH modifier, as long as the pH of the test topical product and the reference standard are well matched. So in conclusion, Q3 characterization of a reference standard provides a profile of physical, chemical, and structural attributes that's quintessentially characteristic of that reference standard. And these attributes may potentially be critical to product performance. Basic Q3 characterization can demonstrate that prospective generic, that test product, and its reference standard are the same dosage form, whereas comprehensive Q3 characterization establishes that detailed profile of measurements for Q3 attributes that may be critical to product performance. And when these are the same for that test product and its reference standard that can support a demonstration of bioequivalence. Now, one of the other things is that it's important to come away from this presentation with clarity about the fact that even if there's no PSG available for a topical product that you're interested in developing a generic for, these generic topical products are generally eligible for in vitro characterization-based BE approaches when two main criteria are met. One, there's no significant difference in an active ingredients or in other aspects of the formulation that may significantly affect local or systemic bioavailability. In other words, the underlying matter is essentially the same. And also that each relevant Q3 attribute of the test product that's been characterized in multiple batches is demonstrated to be within the range characterized for that Q3 attribute of the reference standard, again, perhaps in multiple batches, or at least determined by FDA to be within the acceptable variability for that reference standard. So in other words, the Q3 attributes of the arrangement of matter is also well matched. Now, if you would like to submit an ANDA for a topical product using a characterization-based BE approach, and there isn't already a PSG published, I'd really encourage you to request a pre-ANDA meeting with FDA to discuss your proposed approach. And I'd also encourage that in that product development package, you should include data and information on basic Q3 characterization of the test product and the reference standard product at minimum, as well as product characterizations that are relevant to the nature and the complexity of the product, and would really help to identify potential therapeutic failure modes for the product. With that, I'll say that thank you very much to so many individuals who have helped to establish what I've been able to talk about today, and I really look forward during the coming Q&A session to answering all your questions. Thank you.
Hello, everyone. Thank you so much, Dr. Raney, for the wonderful presentation. I would like to now invite all of our panelists to begin turning on their cameras as I do brief introductions for our panelists. You are able to review the full bios in the resources for this workshop. So on the panel today, we have Dr. Rob Lionberger, who is the director of the Office of Research and Standards, Office of Generic Drugs at the FDA. We have Dr. Pahala Simamora. He is the director of the Division of Liquid-Based Products 2, Office of Life Cycle Drug Products within Office of Pharmaceutical Quality at the FDA. And we have our speaker, Dr. Raney, joining us on the panel as well. So I'll just give it a moment for all of our panelists to join us, and we will dive into the Q&A. Hello. Okay. In the interest of time, we will get started, and I am sure Dr. Lionberger will be joining us momentarily. So thank you to all of you for submitting such wonderful questions. We will go ahead and get started, but please continue to submit your questions because we look forward to discussing them with you. So the first question we have is for Dr. Raney. And there was a question that in our current product specific guidances, there are typically a couple of approaches recommended. One that you mentioned during your talk, the characterization based approaches that are applicable to specific products with a specific formulation, and then often a comparative clinical endpoint study. In your talk, you briefly mentioned that there could be a third category of products that we may be looking at. Would it be possible to provide an example of what kind of products fall within this category and what are the scientific expectations when for establishing BE of such products?
Sure. Thanks, Priyanka, for the Often topical products contain multiple ingredients, and some of those ingredients may exist in the product at a very small proportion of the product, let's say 1% of a particular ingredient. And so when trying to match the composition of that reference product, it can be a very tight target to meet to be within plus or minus 5% of 1% or even sometimes 0.1%. And so there are times when the product is very, very closely matched, but not close enough that it would be within the plus or minus 5% that would fall under the first category where it meets the no significant difference standard. It might be just outside that, and yet it's very close. All the Q3 attributes match, the drug release matches. And so for those types of products, one of the things that we might want to consider is, does that difference, that small difference, maybe it's not plus or minus 5%, maybe it's plus or minus 7% or something like that, does that still qualify as representing no significant difference? In other words, this is not a difference that would impact local or systemic bioavailability. And there could be evidence submitted in the application itself where the PSG might recommend matching the components and composition, matching the Q3 attributes, having an IVRT study, but the PSG may not specifically recommend an IVPT study to evaluate that local bioavailability, or an in vivo PK study to evaluate systemic bioavailability. And by adding on those components and showing that there's no difference by IVPT in local bioavailability and no difference in systemic PK by in vivo PK, then you can demonstrate that the difference in your product is not a significant difference. It's not a difference that affects local or systemic bioavailability. And it's also important to make sure that there's no farm tox issues created by those types of differences, so that it shouldn't exceed IID limits and so forth.
So we will go to our second question now, and this question is for Dr. Simamora. So Dr. Simamora, we received a couple of questions, and since Q3 characterization tests as part of BE approaches are often reviewed by the Office of Pharmaceutical Quality, the first one is, are you satisfied with the current methods and nature of data that are being generated for Q3? And are there opportunities for development of new analytical methods to understand the microstructure better?
It's we do a risk-based approach. I think the same thing like in the product development, you identify the risk and you mitigate the risk. And the same thing with this, especially in the topical drug product world, right? You have something simple all the way to the more complex and the complex one. As the complexity increases and the risk also increases. So the idea is being able to identify and then to de-risk those identified risks, that exactly like what Dr. Raney has been presenting with respect to the quality, the QT characterization test. So some of those examples nicely explained by Dr. Raney, the 10 type tests. There is nothing specific about, what to do. It depends on, case by case, depending on the drug product. And again, like what I said, some of them like quite simple and some like quite complex. As the complexity increase, then, you got more, you need to put, more evaluation to it, more, just trying to de-risk in any potential risk associated with that particular drug product. I hope it answered the question. Any ORS folks would like to chime in?
We actually have a follow-up question. So with respect to the Q3 characterization data that is submitted, what criteria is used to conclude that the formulations are compatible? And maybe we can start with Pahala again, and then others can chime in as well. Yeah, so data is submitted, what is the criteria that we use to say that the products are actually conclude that the products are actually compatible, or that they meet our endpoint that the test and reference products are compatible?
I think as Dr. Raney explained nicely, you have those tests, and we would generate the RLD data, and then you got the variability of the RLD data, and then you conduct your test product. You test the test product, and then as long as that within the variability of the RLD, then I would say then it's the characteristic match the reference product's characteristic. Sam, would you like to add to it?
So once you've characterized multiple batches of the reference product, you essentially characterize what the variability is for each attribute, whether it's the particle size distribution or whether it's the pH. And to demonstrate then that your product matches, you're showing that the pH of your product or the particle size distribution of your product falls within the range you've already characterized. And that's the basic kind of foundation of how the assessment is done. Now, there are some additional considerations that I think maybe are beyond the scope of getting into here, but as an example, part of the reason for looking at different ages of the product is that if you actually know that there's a trend of particle size growth in the product, and your freshly made product is already within the range, yes, but at the high end of what old batches of product is, well, then that may be kind of something that may need to be looked at more closely. So these are some of the ways that the considerations are. I don't know if you want to comment any further on that, Pahala.
We would move on to our next question. And I would also like to take this opportunity to introduce our panelist, Dr. Markham Luke. Dr. Luke is the director of the Division of Therapeutic Performance 1 within the Office of Research and Standards, Office of Generic Drugs. I will share the next question with Dr. Lionberger, and then we will come back to Dr. Luke in a minute. So the question we have for you, Rob, is if a product meets the criteria for establishing bioequivalence using the in vitro approaches, including perhaps IVPT, is there a need to submit information to support the product's in vivo bioequivalence?
I mean, generally no, right? If you provide sufficient evidence for if you think about what the purpose of our regulatory expectations are, right? It's to demonstrate bioequivalence of your product to the reference product, right? Once you've done that, you don't need to, do additional in vivo studies if you've demonstrated bioequivalence by an in vitro approach, right? But it's the goal of the scientific goal of our review of your application is, have you demonstrated bioequivalence, right? Whether it's by in vitro studies, whether it's by a waiver, whether it's by in vivo studies. And I think one important aspect of this whole enterprise in the topical area that Sam and Priyanka and Markham have been leading over the past years has really been providing in vitro ways to demonstrate bioequivalence. And starting first with formulations that are very, very similar in composition, right? But recognizing that the in vitro approaches and the situation that we're in now is these in vitro approaches are very powerful. They characterize the physical attributes of the product. And these physical attributes of the product are things that affect how the patients perceive the product as well. So it's a very important part of the topical products in themselves. And so as we move to this approach, now we're recognizing that, again, the power of these characterization approaches is not just limited to you're exactly the same in your composition. Like as Sam very well mentioned in his presentation before, right? Sometimes you have an inactive ingredient that, is there at a very, very small amount, right? And it has, it may have some purpose there, but that purpose does not affect the bioavailability of the drug or the perceptions of the product. It may be a preservative to ensure stability, and we test stability, and the product stability is okay, so the differences in a preservative are acceptable because they serve their function, but the rest, but it doesn't limit or affect the rest of the availability of the product. And so I think that's where we are in the evolution of our scientific framework around topical products, is really recognizing that we can go beyond, you have to be exactly the same in your inactive ingredients to use the characterization approaches, to say that based on our scientific and understanding of the formulation science of these semi-solid dosage forms, there's a broader range of differences that still fit into this in vitro approach framework. And I think that's why we've articulated, no significant difference, right? It means, look, I found I have a difference in composition that's there, but I've looked at all the evidence, and the in vitro evidence is sufficient to say this is, and our understanding of what that component does in the formulation. That's another aspect of this, is that as we develop our understanding of the formulation science, and you present this in the application, it says, what is that purpose of that difference, right? That helps us make this assessment. Does this difference, in fact, the bioavailability of the drug or the clinical acceptability of this product? And we put that together. And so this is an ongoing process of expanding the space where you can reliably use the characterization-based approaches to bioequivalence. And I think that's evolution. We've made a lot of progress in the past 10 years of really saying, look, we're routinely improving topical products based on the Q3 approaches. And now we're really looking to say, well, look, as you go to the edges of this, you might have fallen outside of our Q1, Q2 rules. I can't use that. Recognize that the science doesn't support that, right? They're scientifically valid approaches. And that's really what we're talking about in this webinar, and some of the research that we have ongoing is understanding those differences so that we can expand the use of these in vitro approaches in the topical space.
Thank you, Rob.
And but I mean, certainly at some point you may change your formulation so much, or you may look, I measured something about my product, it's different. In those cases, we might need more data. You might need in vivo data. Sometimes it might be, well, in this case, you need an IVPT study that's trying to mimic the interaction of the formulation with something like the skin, and that's the additional piece of information you need. So as the differences, either in the formulation themselves or the measured properties of that formulation get bigger, then there might be different types of evidence that we need in order to understand the sameness of the product. And we continue also to work on our understanding of the formulation science, as well as the models that can help predict this. And that's also a very helpful part of saying, look, I changed this physical attribute. Is that really expected to affect drug delivery? So you can use a modeling approach to sort of say, look, I've changed my viscosity by this small amount. Is that going to change the drug transport rate? And understand that better. So there's a lot of ways that these all fit together into this very significant and very successful effort that we've been moving in the topical area over the past sort of 5 to 10 years.
Thank you so much, Rob. I think related to what Rob just said, Sam, we have a question for you. So the question basically says, to avoid issues during the actual ANDA review process, can we submit a control correspondence in case we have to use an excipient with slight differences compared to the reference product?
Development meeting process may also be an ideal way to be able to provide information about both at minimum basic Q3 characterization, but as much in characterization as is relevant to the complexity of that product. and then to also be able to explain the thinking behind what you have assessed to be the failure modes for BE that might arise from using that different excipient, and what you proposing to utilize as the evidence to mitigate the risk of the failure mode associated with that change at the excipient. So often a pre-ANDA meetings process can provide a better forum for discussion and a little bit more in-depth dialogue surrounding that.
Thank you, Sam. The next question we have is for Dr. Luke. And the question is, what are your views regarding the effects of Q1, Q2, and Q3 differences on the safety profiles, and how should they be assessed within the scope of our BE approaches? We can.
Rob talked about this very nicely. I think that this is the Q3 concept started about 20 years ago, and since then we've put looked at these things from a variety of ways. Like, is it lotions, creams, ointments? But then we're actually categorizing each individual product itself, because two lotions may be different, two creams might be different. And in that context of the safety piece, we're looking at not just whether it's a gel, cream, lotion, for example, but also where it's applied, and what the importance is for certain Q3 aspects. The rheology, for example, could be more important for a product applied to the face. And so there's a certain issues about how the runniness, does it get into the eye, things like that, and other aspects to the product. And so these are important things that we learn about specific topical products. Hence the notion of product-specific guidances for each of the products that we develop. And like Sam mentioned, the importance of pre-ANDA interaction for specific products that companies are trying to develop. Should they be non-Q1, Q2, and we're looking at different excipients with perhaps slightly different profiles, both physical, chemical, or certain considerations for impurities, etc. And Pahala also touched on some of those as well.
Thank you.
Does that answer the question? Yeah, thank you.
For understanding the Q3 profile, but sometimes it is not possible to procure multiple lots on the market. Can you suggest what approach should be taken in those cases?
It could be one. I think the best thing to do is to have a discussion, to communicate with the FDA, and then have a discussion that way. That's all I can provide for for that question.
Thank you, Pahala. The next question we have is for you, Sam. The question is, which is more important to focus on in an ANDA application for topical products, basic Q3 or comprehensive Q3? And then there is a related question which basically asks, like, when our guidances say that to be able to evaluate a product, Q3 characterization should be done, and it doesn't mention IVRT, do those guidances actually require IVRT studies? And I think we are mostly referring to guidances for AT-rated products in this question.
reference product itself, that at the very least, any product developer, any generic product developer would want to conduct basic Q3 characterization of the reference product, and equally to have the most minimum product and process understanding for your own product, that you'd want to have basic characterization of your own product as well. And certainly for AT coded products or AT rated products, that basic Q3 characterization is a foundational way to be able to assess and compare the dosage forms for those products. And if IVRT is not recommended for that product, as for specifically for AT coded products, many times IVRT is not recommended, then it's sufficient to follow the recommendations in the guidance for those specific products. Now, the comprehensive Q3 characterization is particularly meaningful when you are not using, for example, a comparative clinical endpoint bioequivalence approach. In other words, the approach to demonstrating bioequivalence is now going to rely upon demonstrating that the prospective generic product is as identical to the reference product as batch to batch of that reference standard is to itself. So these characterization-based approaches are when that comprehensive Q3 characterization becomes absolutely essential. And as you've seen, there are PSGs that will say specifically characterize ABC. When a PSG is not available, now you have a good understanding of which of those characterizations are going to be relevant as part of the comprehensive Q3 characterization for your particular product.
Prospective generic product should be Q1, Q2. Can we develop a product that is non-Q1, Q2?
Right, for topical solutions, FDA's regulations are very clear, right? Topical solutions are not required to be Q1, Q2 to their reference listed drug or reference standard. I mean, Sam touched on this issue in his talk as well. As you think about our system, however, for the topical solutions, right, if you have a Q1, Q2 solution, right, you might be eligible for a waiver, right? If your formulation is different, then you might have to show additional data to show that your topical solution is going to be bioequivalent, right? Because there's things you can change in the solution, right? I can make a solution, if I put some different excipients, I can make that solution much more viscous. I can even affect the drug delivery, right? So topical solutions, even though when they are Q1, Q2, we generally will give a solution type waiver. Really don't need to do significant characterization or comparative work around that. Although I think generally it's generally good practice to measure all of the properties for even your Q1, Q2 topical solutions as part of your pharmaceutical development. But as you deviate from the Q1, Q2, right, then it becomes a question, at what point is the characterization approach insufficient to demonstrate bioequivalence? And we need to have some sort of data that either is mimicking the in vivo situation, like an IVPT, or some type of in vivo study, going beyond that. For example, like hypothetically, I don't know why anyone would do this, right? But you might add a penetration enhancer to your topical solution. And when that comes to FDA for a review, right, FDA reviewer would say, well, wait a second, you added a penetration enhancer to your topical solution. Maybe I'm not going to give you a waiver of that, right? Maybe I need more data to say that your change in formulation did not affect the bioavailability. And to go back to the RLD versus reference standard for this, right, again, in the topical space, different than, parenteral products, there's not a regulation that says you have to be Q1, Q2. So if FDA has, if the RLD is not available and FDA has demonstrated a reference standard, right, that reference standard is the product for which you should demonstrate bioequivalence. And that comparison might be based on how different you are from that reference standard. So if that becomes the bioequivalence standard for this product, then that's what you'd have to do. And if hypothetically you made a big enough change that you decided to do an in vivo study, you'd use the reference standard in your in vivo study. And that's why FDA will designate reference standards for these products, even though, for a solution product, it might be very similar. And certainly if you'd say, well, look, I know that some cases FDA's reference standard might not be Q1, Q2 to the original RLD, right? And that's the space where you might want to, discuss in a particular control correspondence or meeting exactly what that means. Maybe there's something that's information that says, well, look, this difference in formulation does not seem to have a significant effect on bioavailability because FDA approved both of these products. So it might be you might want to clarify exactly what the best pathway is there if you're trying to say, well, look, I'm similar to a discontinued RLD and not the current reference standard, and what the appropriate approach is in here. And again, each different route is different. In the parenteral products where our regulations talk about Q1, Q2 sameness to the RLD, you always have to reach back. In the topical products, there's not a regulatory requirement to reach back, but the comparison is part of the bioequivalence bridging to FDA's designated reference standard. But there may be some places where you're looking at that, that understanding about what the characteristics of the original RLD were might be a valuable piece of putting together what type of formulation is acceptable and what types of differences are significant in a particular case.
Like a one species or two, right? It's a mixture. And then as Sam explained in one of his slide, especially the one that has a lot of this petrolatum in the formulation, 70-75% plus. Now the behavior, the characteristic of the products, the majority is being contributed by this petrolatum. So it may be a good idea just to see, because there are multiple suppliers that come from different sources, and different fraction, different hydrocarbons fraction. It would always be a good idea, and also part of the product understanding, to have that characterization, to be able to know exactly the product, what we have, what the RLD have. Will it make any difference? Maybe not. But if we know and we studied well and we know the nature, the product behavior, and we know ours as well, right? So I think that would be making a good story, for the agency to during the review. I hope it helps answer the question.
Thank you, Pahala. Sam, would you like to add anything to that?
It's not one. It has it's a multi-excipient excipient, so it's quite complex, and that has a lot of molecular species in there. And so you can certainly use tests from the USP monograph, and those are specific tests that you can run. And so, for example, there's a test where you end up boiling the petrolatum and looking at some of the organic acids that come out, and you can do a colorimetric test on essentially the pH of that solution. So while the USP monograph may describe it that way, for more precision, you can just use a calibrated pH meter and actually measure that. You can do a lot of those kinds of tests and compare them for the product made with your source of petrolatum compared to the reference standard product. But then also you can essentially use, let's say, for example, GC characterization of the relative proportions of each of the individual species, which on a plot would basically show up like almost like Pahala's bookshelf. They're very nicely organized with books, and you'll see that there's sort of a profile to the enrichment of different species of hydrocarbons, and you can compare the profile of that distribution of different hydrocarbon species, and you might find that they're relatively well matched, and that in one region in your product there's a real enrichment in these, let's say, C8 to C10 hydrocarbons, which may be permeation enhancers, that could really affect the bioavailability of the product made with that source of petrolatum. So it's actually much better to be able to find petrolatums that match across the entire range of hydrocarbon species characterizations.
Thank you, Sam. One last question before we end this session, and I'm going to direct this question to Dr. Luke. So the question is, if additional experiments other than those listed in the PSG are conducted to support Q2 differences or Q2 that is slightly outside the range, does it have to be discussed through a pre-NDA meeting?
Developing that product, that they do come in from what is listed in how it's described in the PSG, in the product specific guidance, that they do come in and meet with us. And of course, it depends on how much of a difference it is. And we're open to you suggesting a pre-NDA meeting, and we will tell you whether it's appropriate to have a meeting on this, or if it could come in as via some other method of discussion. But ideally, a pre-NDA meeting would be the best way to have the discussion about an alternative method to what's in the product-specific guidance.
Thank you, Markham. Okay. With that, thank you so much to both Dr. Raney for the wonderful presentation and